model study was prefaced on this idea that we know that cells in the brain are not sort of static. They're not just two-dimensional things. They're constantly moving and they're changing and responding to the environmental stimuli around it. And so basically these experiments were born out of this idea of, does chronic migraine alter that in any way? Because it's such a sort of fundamental question that we kind of wanted to just do something very basic and see, well, what happens when we use a preclinical model of migraine? Is that going to alter the way that these neurons actually move and change and their ability to respond to their environment? Basically, what we found was that in a model of chronic migraine in mice using nitroglycerine, which is a known human migraine trigger, we found that in certain head pain processing regions, such as the trigeminal nucleus caudalis, periaqueductal gray, and somatosensory cortex, there was decreased neuronal complexity in this model. And to follow up on that, we also looked in a brain region such as the thalamus, which is an obvious known relay circuit for pain processing. And in that region, we actually found that there was an increase in neuronal complexity, suggesting that there seems to be this disbalance that's happening between these brain regions, and that this might be some sort of signature of migraine chronification. We looked in regions that were sort of more peripheral to head pain, so things like the amygdala or the caudate putamen or the spinal cord, and in those cases, we found no change in neuronal complexity, again, suggesting that this isn't something that's affecting the whole brain. It seems to be more affecting these pain processing regions. We also looked in another mechanistically distinct model of migraine, which is cortical spreading depression. And in this model, we found that similar to the nitroglycerin model, there was decreased neuronal complexity in the somatosensory cortex, in the trigeminal nucleus caudalis, and the periaqueductal gray. And this is important for a couple of reasons. One, it shows that in these two distinct models of migraine, we see the same types of changes in neuronal complexity, and that is a decrease in these three regions. The other thing that's interesting about it is that cortical spreading depression is something that really is evoked on the cortex. And so and there's often been a lot of questions about whether cortical spreading depression or depolarization might then be activating pain networks in other parts of the brain. And these studies suggest that, yes, in fact, they sort of corroborate other evidence to show that trigeminal nucleus caudalis and the periaqueductal gray are also affected by something that's happening at the cortical level. And then the final piece of this is that we compared and contrasted with a model of complex regional pain syndrome, which is totally different from a migraine model, but still has this sort of chronicity of pain. And there's this sort of movement from in complex regional pain syndrome from early stages where the mechanism seems to be more peripheral to later chronic stages when the mechanism seemed to be more central. And what we found there was that in CRPS, that there was also changes in neuronal complexity, but that they were different from the ones that we observed in our migraine models. So in this case, we saw an increase in neuronal complexity in the periaqueductal gray, whereas in the migraine models, we found a decrease. And then we also observed a decrease in neuronal complexity in the hippocampus, which, is postulated to be part of that sort of brain fog that accompanies CRPS. So basically what we found is that it seems that alterations in neuronal complexity are hallmark features of chronic pain states, but that depending on the chronic pain state, there might be very different cytoarchitectural signatures associated with them.